So again, what is perhaps the most important aspect that we had alluded to on Monday? We discussed it in depth yesterday, and we begin with that today. Okay? When it comes to dietary proteins, how do we break them down? And we need to start with the stomach and end with moving into the small intestine. So what's happening? Today you get your mini corn dogs. There's proteins in there. How do we break them down chemically? In other words, there's something happening inside the stomach. What is that? You answered it yesterday. You can probably answer it again today. So my point is we So with that, what's happening inside the stomach? Give me a couple cells. Okay? Okay, did we catch that? Okay, so what happens is you have this bolus of food, which is a chunk of material entering into the stomach, and there's three types of cells within the lining of our stomach. We have mucoid cells producing mucus. We have chief cells, which then secrete the pepsinogen, which is that active or inactive? It's inactive until the parietal cells secrete the acid. And once those two come in contact with one another, the pepsinogen is then activated by the pH of the hydrochloric acid, making pepsin, which is then a universal enzyme that breaks down dietary proteins. Now, there is not one fix-all enzyme for digesting protein. We also have enterokinase. That's a secondary enzyme. And we can see it works similar to the pepsin mechanism. Okay? So what that means is you also have trypsinogen, which is inactive. Trypsin activates it to enterokinase, which then also breaks down proteins. Okay? The pancreas also secretes, yes? No. We want to know that trypsin activates trypsinogen to enterokinase. Yes. I don't know why it's not called the same as that of pepsinogen and pepsin, but it's a mechanism that works similar to that of the pepsin mechanism. That is correct. We do need to know where the pepsin is coming from, and that, and of course, is within the stomach. Okay, the pancreas gets in on this too with two different types of enzymes. Okay, one breaks down carbohydrates, so that would be pancreatic what? Pancreatic amylase. Now, we said yesterday the reason we call it pancreatic amylase is that's not the only source of where that enzyme is found. Where else do we find it? In your mouth. That's right. Okay, so what is another enzyme secreted by the pancreas? Lipase. In that, lipase is responsible for breaking down what? Lipids. And this is what we're talking about, yes, right here. One of the things we discussed yesterday. Okay, so we move on with the liver, and probably, of course, every organ that we have is going to be important, but pancreas does its assorted functions, the kidneys does its assorted functions, not with digestion, more so with excretion, but the liver does so many metabolic functions for us that without a doubt probably and it's difficult to live without any three of those, but not you can say one's more important than the other, but the liver certainly does so many actions for us that it would be difficult, and people do have a hard time living without their liver, or without a properly functioning liver, is what I'm trying to say. So what we see happening here, okay, you might be thinking, well, it's talking about a normal blood sugar level. You might be thinking, well, why are we saying the liver does that when the liver is not what secretes this hormone? It comes from the pancreas instead, and that starts with the letter I. It's the insulin, okay, that helps maintain this blood sugar level, the hormone causing the liver to do this, okay? So blood glucose levels are monitored by, of course, your brain or your central nervous system, but it's the hormones secreted by the pancreas that causes the liver to do one of two things, okay? If there's too much sugar in our blood, if we need to lower that, one of two things is going to happen. Either one, start taking a bunch of these monosaccharides of glucose and linking them together to form glycogen. That's one way. And another way is causing the cells inside of our body to burn that sugar. That's one of two ways that blood glucose levels can be lowered. Well, maybe we're a little sluggish and we've got some excess sugar. Also, what can happen, we need to raise it. Well, you have this excess glycogen in your body. The liver will then metabolize this glycogen back into glucose. And one way to look at that is like this. Okay? Hopefully it'll make more sense in this fashion, is each one of these, just picture these individual links in a chain, okay? These are all glucose molecules moving around in the blood, okay? So having, is there 7, 2, 5, yeah, 8, okay? We got these 8 molecules, that's too high. We need to lower that down to maybe just even one. This is just one segment or one drop of blood in the entire body. Of course, there's going to be more than this. But we need this to be lowered. So one of two things is going to happen. The liver may be stimulated by the pancreas, okay, to say, hey, we need to lower our blood sugar levels, and we're going to start linking these together. Okay? And once you start consolidating or pulling these individual molecules from glucose into glycogen, blood sugar levels will start to go down. And that's sometimes what the body senses you need to do. Okay? But just in the opposite way, then, we can also have a stimulus say, hey, our blood sugar levels are too low. We need to start breaking these apart, this glycogen, back into individual glucose molecules. So this is one way to look. It's kind of like a reversible reaction, depending upon what it is your central nervous system or your brain is sensing. Okay? Just like if you feel cold, you go to the thermostat, you turn the thermostat up. Okay? It goes past the set temperature. Okay? Then the heat kicks in, so we get more heat. More heat in this direction. Now we're getting kind of hot, so we turn the thermostat down, so all these heat molecules, we start putting them back together, so we start to cool off. Okay? Now, this is pretty higher order thinking. This is maybe a hard concept to get down, but the reason that we go and discuss this in this level is I'm talking to you and discussing this in a certain manner because chances are you will also be discussing this in a certain manner down the road, whether it's up at SDSU, University of Kansas, wherever. These concepts are vital to get through anatomy and physiology class. Okay, let's see here. We want to make sure that we can finish this tomorrow. We've got 10 minutes left, and okay, let's maybe do one. Okay, just 2 more slides and we'll be done. Actually, we'll stop after this slide because I guarantee you, you're going to have to be able to describe on your exam the pepsinogen mechanism, also why the liver is so important and its vital function dealing with proteins. And this is it right here. Because what happens, what the liver does for us with all the waste products that may be produced, uric acid, okay, is broken down into something that's water soluble, urea. So now we can get rid of that in the form of urine. Once it becomes water soluble, it then gets sent to the kidneys, then to the bladder, and then out the body. Okay? So as you can see, protein metabolism, waste removal, and also creating clotting factors. Those are the 3 very important functions of the liver. Okay, does everyone have this information? Okay, so you've got the remainder of the time, work on your journals, work on your papers, whatever it is you feel is the most beneficial for yourself. Okay, and we will pick up with this again tomorrow, and also move forward. It says diagrams. Those are the same diagrams that you will be responsible for next week. Because as you could tell on your vocab portion that you just had taken, how many different types of ducts are associated with the junction between the liver, the gallbladder, and the small intestine? How many different types of ducts? There are 4 total, but do some of them, are they named the same? But yes, you're right, okay? We could say left and right hepatic duct, which would count as 1, you could say, because it's a left and a right. You also have a cystic duct, that's 3. And then where the hepatic duct meets up with the cystic duct, you have the blank bile duct. That's the fourth one. Common bile duct, that's right. So you have your hepatic ducts, again, that come down, meet the cystic duct from the gallbladder, and they form a tributary of one another. Common bile duct then is then dumped into the small intestine. Okay, we will continue with this again tomorrow.